start learning the new topic called as the sex-linked inheritance. Have understood? See here, what is sex-linked inheritance? The appearance of the triad, which is due to presence of an allele. Yes, what is allele? See, last class I taught you know about the detailation of an alleles. Alleles are nothing but there is a pair of characteristics, the contrast characteristics that produces a different effect. See, like XX in a case of a female and X and Y in case of a males. So that produces different effects. Yes or no? Those alleles exclusively either on the X chromosome or the Y chromosome. Have you understood now? In such cases are called as the sex-linked inheritances. In that sex-linked inheritance, X-linked inheritance is the most important, the subtopics. The due to heredity. What is heredity? The heredity, which is nothing but the transfer of characteristics from the parents to the offspring. Yes or no? In such cases, there are some genetic disorders that may the transfer through the genes. Yes or no? From the grandparents, it can be the grandparent, or the immediate parents called the father or mother also. There will be chances of receiving some of the genetic disorders. Yes or no? In such cases, there are mainly the two diseases. They are the major diseases which can affect to the newborn babies also. Have you understood? In the major two diseases are the hemophilia and color blindness. These are the major two the genetic diseases which can transfer to the parents to the offspring. Clear now? But here the most important thing is these two diseases, they are more common in males than females. That you have to notice here. That is the most important. These genetic diseases, they are more common in males than in females. You know the reason? Well, the reason is the recessive gene occurs on X chromosome. The recessive gene they occurs in X chromosome, because of that females they will be having the X and X, double X chromosomes. Yes or no? But whereas in case of a male, X and Y. So here you can notice that the recessive gene occurs in X chromosomes here. Have you understood? So that is the reason why the genetic diseases like color blindness and also the hemophilia, they are most common in males. So let us start with the first disease called as color blindness. Color blindness, inherited diseases. Yes, of course, it's an inherited diseases which is affected due to the individuals and cannot differentiate certain colors. I think already you have seen and you have noticed some of the people who cannot differentiate certain colors. Have you understood? In such colors, majorly, which are those two colors? The red and green color, they are the two major colors. It's very difficult to differentiate in the color blindness, those who are suffering from the color blindness diseases. Have you understood? The rest of the colors they can be easily identified. But here, the red and green, it's very difficult to identify, whereas in case of the color blindness, the persons. And one more disease called as hemophilia. Hemophilia means it's like genetic diseases, hemophilia. That genetic diseases, that risk of bleeding to death in the sense, see some people, it's a very rare cases. So that is why it's a very dangerous thing. Which one? The hemophilia. There are some people, if they met an accident, forget about the accidents. If the minor accidents or minor injuries also, once the blood starts bleeding, it's very difficult. And it is not easy to stop, the clotting of the blood, it takes time, In such period, in between that period, the person may lead to the death also. There will be more chances of a death of a human being, whatever it is, those who are suffering from hemophilia. So, let's have some, the problematic, how the progeny is, if in case, the problems. When in case of the normal mother, when the normal mother, they married with the colorblind father. So what could be the progenies? So what could be the next generations? So let's have here. As the last class I told you that, when you are calculating the genetical problems, it's easy to calculate it with the help of this checkerboard. This is also called as checkerboard. The differentiation of the triads and also different gametes. We can use this Punnett box, checkerboard, we can say this one. Have you understood? So let's have see, even in case of normal mother in the sense, you know that in all the ladies from the baby girl to the till the human, the normal mother in the sense X and X chromosome. They will be having two X chromosomes. That's why we call it as a recessive chromosome. Have you understood? And here, whereas in case of color blindness in the father means they will be denoted as X and Y. But have a look here, on the surface of an X, you can see that the immediate action over there. Yes or no? So let's have a calculate the normal mother and the colorblind father. So the new generation, the progeny, what could be the possibilities? Let's have a look here. When the first generation, X and X, here you can see this is colorblind father. Whatever the colorblind father, we have to notice on above the surface, and we have to notice in the another surface of the checkerboard or a Punnett box. Clear now? When you calculating the triads, when you multiplying the triads, you know how to like this. From here, X and X, we can see this one, X and X, here the colorblind father. These are the triads from the colorblind father. So that is why colorblind daughter here. The carrier daughter, colorblind daughter in the sense carrier daughter. So there will be chances of visibility. The daughter can be visible. They can identify the colors easily. So once the age passes, there will be chances of the colorblind also. Understood? And the next one, X and Y, which indicates that it's a normal son. There will be no chance of the carriers are they suffering from the color blindness. Here the second generation is called the normal son. A third one, X, again you have to multiply with this one. This one X to the same. How you calculated in the first generation, the same way in second also. X and X. Here also the same thing. The carrier daughter. And one more, X and Y, it's a normal son. So there will be chances of the next generation, there will be chances of like 50-50. The 50, the next generation might be suffering from the color blindness or a carrier daughter or no. If not, there will be chances of the normal son or a normal daughter or carrier daughter. Have you understood? So have a look here, X and X. The heterozygous, you can see this one, this is a normal, this is not in a normal. They are not in a homozygous, they are in a heterozygous. Heterozygous, the dominant, they are in normal vision. So that's why we call it as always the first triad. This is the first triad, yes or no? This is the first triad. This X which decides the possibilities. So that's why we call it as the dominant, and there will be chances of the normal vision. There is no carriers in the data. Have you understood? And X and Y, you know very well about that. If there is no indication of the carriers or the color blindness, there will be chances of the normal son. And have a look here in a second case. In a second case, the carrier mother, when the mother, she has the carrier mother and the normal father. In the first case we have studied the colorblind father, but here there is no the colorblind or there is no diseases with the father, whereas in case of the carrier mother, let's have a look here. In the same way how you calculated here, the same you have to do this one. X and X. You have to write in a Punnett box. This is normal daughter, and X and Y it's a normal son. X and Y it's a normal son. The next case X and X, here the carrier daughter. Of course, there will be chances of the color blindness, not immediately. Have you understood? That's why we call it as a carrier daughter. But you can see here the normal daughter. Yes or no? And the next one, color blindness son. So this is how the next generation or next offspring there will be chances of that could be carrier daughters or color blindness son. But here in the case of case 2, you can see there will be 50%. The one child the born in a normal and a healthy condition, one girl baby they are born in a normal condition, and one girl baby there will be chances or possibilities of carrier daughter, and one boy baby there will be chances of color blindness son here. And whereas in case of in your textbook they have given that the third problem in a third case, have a look here, the carrier woman and a color blind man. Here we have seen that first case the normal mother and a color blind father, second case carrier mother and a normal father, the third case carrier woman, which means there will be possibilities of the mother also they may affect it to color blindness and in case of father also he was affected from the color blind. So let's have a look here. What are the progenies? X and X, they both are the color blind, the daughters. Have you understood? And X and Y, the color blind son. X and X, carrier daughter here, and here the colorblind daughter. Have you understood now? It's a domination. This gene which is dominated are suppressed by the another genes. Yes or no? So that's why there is colorblind daughter. But whereas in case of third generation, the third generation there will be chances of the carrier daughter, which means the child has to be normal in condition. The third, fourth generation X and Y, there is no possibility of the newborn, the boy baby will not suffering from any kinds of diseases. So which is nothing but the boy baby is a normal in conditions. Have you understood? So this is about the sex linked inheritance. So how the suppressive or recessive genes which mainly which indicates the future or offsprings of the possibilities. And still there are some the experiments on the different plants and also and some of the human beings also. Now depends upon the genetic disorders and all that. See the basic fundamentals. So before I told you some of the genetic disorders, genetic diseases. Yes or no? The Mendel was a famous scientist who mainly worked on the plants. Yes or no? To get different varieties of yields. Yes or no? See what we call nowadays hybrids. In that hybrids, the different varieties, the variations, the mainly who shows the scientist, the Mendel. He was the scientist who worked on the different varieties of the plants, but to successfully succeed are the pea plants. The pea which is nothing but you can see that. So the pea plant, the botanical name of the pea plant, the Pisum sativum. So the binomial nomenclature already was studied in the 9th standard. And that is the lesson. Is or no? The Pisum sativum is the botanical name of the pea. See what it does. See George, the Gregor John Mendel, you also know exactly. This is the original name of the Mendel. Gregor John Mendel. He was an Austrian. He was a scientist. We understood. So he mainly worked on the pea plants. mainly the three reasons why you have selected the mainly the pea plants, why not in other plants? Yes or no? So you would have selected the different plants, pumpkin, whatever. There are so many there. But he selected the pea. So mainly the three reasons. The first reason is the many varieties were available in alternative form of characters. When you cross in the first generation with the second generation, or when you self-cross the first generation, you may get the varieties. The continuation of the generation also is in most of the cases. But when you artificial crossing, when you do the artificial crossing or artificial the pollination, you may get the new varieties. That is the one reason he has selected the pea plants. The second reason is pure forms that breed true. We will get the pure forms. You know why? The peas are normally self-pollinated. Yes, you know that what is self-pollination. Yes or no? The pea plants also are the examples of self-pollination. They easy to remove the reproductive parts. You know the reproductive parts of the flowers. Yes or no? The anthers, the male gametes. The male reproductive organs are the anthers and the ovaries are the female reproductive parts. Yes or no? But here, in case of a pea, it's easy to remove the reproductive parts so that instead of the self-pollination, you can do the artificial pollination. So that will help you to get to know the new varieties are a continuation of the same kinds of varieties when you do the artificial pollination. Understood? So that is these are the three reasons why he selected the pea plants as the experimental purposes. In a pea plants, again, he has selected the pair of the contrasting characters. He is not selected in all the type of characters present over the surface or inner surface of the plate. But the mainly is selected the 7 pairs of the contrasting characters. So what are those 7 pairs of the contrasting characters? In that, the first pair of the character is the flower color. You have seen that, pea plants, the flower color. There will be different types of colors of them. Sir, no? In that mainly, you selected the color, the flower color. That is the purple, it's one type of color, and white is another type of color. And seed color. The seed color also you might get changes, right? The seed color, yellow color and also the green color. They will do differently. Why? Because of when you self-cross F1 generation, you might get the same varieties or the different kinds of varieties in the F2 generation. Understood? So that we will study one by one now. And the seed shape, you can see some of the seeds, they will be having a different shape if they belong to the same family. Understood? If they belong to the same family also, the shape of the seed will be quite varies each and every the species. Understood? In that he selected the seed shape, the round and the wrinkle. The next one, the pod color, whatever we can call, we will call it as the fruit, no? Yes or no? That fruit, the pod color, that's a green and a yellow. And the pod shape, you can see that the pod shape sometimes the inflated, there will be no, the various no like the constrictions, there will be no constriction, the inflated, there is no external surface is a normal in condition. That's we call it as an inflated and a constrictions. For example, you can see the constriction groundnut. You can see that wherever the seeds are present, there will be a small grooving lines. That we call as a constriction. And the inflated, there is no constrictions over there, it's flattened. The next one, the flower position. Flowers appears in different parts of the plants. Yes or no? They mainly the flower positions. He has selected as axillary. Axillary in the sense the nodal regions. Axillaries and the terminal, the tip of the flowers. He has compared, is selected two different the pairs of contrasting characters. And the next one, the plant height. He has selected two different types of plants. The one plant that would be the tallest, the one plant that would be the dwarf, the short plants. So these are on the basis of all these 7 contrasting characters, you work, and mainly succeed to get to know the new varieties and also to how to maintain the same varieties for the next future generations also. Have you understood? So let us start with the first case, monohybrid cross. Monohybrid cross, when you elect the single character, when you elect single character, that's called as monohybrid cross. In that monohybrid cross, the terminal flowers and the axillary flowers. See, you know the terminal flower, the triads will be denoted as the small and the axillary flowers they are denoted as capital A. When you have the homozygous pair of the triad, you can take only the the triad. Have you understood? You no need to take both the two letters. Whatever you have the same type of letters. Have you understood? So if you take this, when you cross the first generation or when you cross the terminal flower, the axillary flower, you will get the first generation, the capital A and a small a, which means this the capital A which dominates the next character, that is A, the capital small a. So it shows that, see, whenever you have the cross with the different types of plants or whatever it is, the first letter, the first triad will indicate the height, whatever the position, whatever the color, that's mainly that depends upon the first letter of the triad. So which means the first generation, the all are axillary flowers. It's a completely dominated by the to the terminal. There will be no expression which completely suppress the characteristics of the terminal flowers. In F1 generation, you will get all the axillary flowers only. When you self-cross F1 generation, when you self-cross the F1 generation, let's have a look here. So how to self-cross? You have to take the same, these triads. Self-cross means, again you have to write another triad. The self-crossing of F1 generation, the capital A. This is how you have to write the separate, the triads. Have you understood? Capital A and a capital A, capital A and a, small a and a capital A, and a small a and a small a. So let's have a look here. You can see three different plants, that produces axillary flowers. Yes or no? The three plants, they produces axillary flowers. The only one plant that produces the terminal flowers. So these are the morphological, which means externally these characters are visible. Have you understood? In such characters we call that the phenotypic. Phenotype, that is 3:1. 3:1. They are called as the phenotypic. Whatever the visibility is. Understood? In such characters we call as phenotypic characteristics, phenotype, and genotypic. Genotypic means, we'll have a look here. See here, they are completely axillary flowers here. Yes or no? That means it's a one in axillary flowers, the completely, and there will be two plants that suppress the terminal flowers. So that's why we call that 1:2, 2:2, and there will be one more, one plant that completely bears only the terminal flowers. This is called as the phenotypic ratio and the genotypic ratio. Have you understood now? Genotypic means, see, whatever the genetic possibilities are there, is such called as genotypic. Phenotypic means the external visibilities, whatever we can see is that the flowers, easily we can identify, but we can't identify the genotypic ratios. Yes or no? The monohybrid cross, the phenotypic ratio is called as 3:1, and genotypic ratio of monohybrid cross we called as 1:2:1. In the same way, if you take The case 2, for example, the tall plants and the dwarf plants. See, whatever you do, the exact procedure only you have to follow in all the 3 cases. For example, you have a look here, the tall plant and the dwarf plants. The tall plant and the dwarf plants. As I told you before, when you have same type of characters, you can take only the single characteristics. Understood? Which means the F1 generation is a completely which produces tall plants. It completely dominates the dwarf plants. When you self cross these tall plants, so how we will get the same kinds of F2 generation also. When you self cross this one, capital T, small t, capital T, small t. Have a look here. Capital T and capital T. Capital T and small t. Small t and capital T. The last one small t and small t. So you can see the same kinds of F2 generation, whereas in case of the case for monohybrid cars, that's a terminal flowers and also axillary flowers. Here also the same. The F1 generation which completely dominates the dwarf plants. Only the tall plants will express in a F1 generation. But whereas in case of an F2 generation, when F1 generation self cross the F2 generation also, there will be possibilities of the most of the plants will be in a tall. Most of the plants will be in a tall, which means it's the phenotypic ratio, that's all 3:1, and the genotypic ratio 1:2, 2:1. But the last, in case of case 3, you can see that different types of characters we are taking. But the following, all the problems, how we are going to solve, the same procedure we have to follow. The triads, only we are taking the characters only the different when it comes to the monohybrid. This is all these cases we are discussing only the monohybrid, not the dihybrid crosses. Have understood? We are taking only the single characteristics of each plant. Here have a look. In this case also the same thing. Capital R and small r. When you self cross this one, when you self cross, what you will get? Capital R and capital R, capital R and a small r, capital R and a small r, small r and a small r. So this is you will get the F2 generation. So here also the same thing. The phenotypic ratio, that is 3:1, and a genotypic ratio, that is 1:2:1. So this is how we have to calculate the monohybrid crosses. In examinations also, if they give single characteristics, you have to go through the monohybrid cross. If they will give 2 different types of characteristics, that you have to be considered as a dihybrid cross. You have learnt how to solve. The Mendel has selected only the single characteristics from the different plants. But here he was tried somewhat differently. So the dihybrid cross, the breeding with the pair of the different characters in the different plants. When you cross, so have a look here, the breeding results with the 2 pairs of contrasting characters. The last problems we have solved that only the 1 pair of contrasting characters, such characters, tall or a dwarf, axillary flowers or a terminal flowers, yes or no, wrinkled or a round. But here the dihybrid means the result in the 2 pairs of contrasting characters here. See how to solve these, the 2 pairs of contrasting characters. So let's have a look here. What are the 2 pairs of contrasting characters here? The round yellow, it's in 1 pair of characters. The round yellow, which is denoted as the capital R and the capital Y and capital Y. Wrinkled green. See, when you have selected as in a round yellow, you will be denoting as in a capital, the capital letters, yes or no? But these are the different, wrinkled green. So that it doesn't mean that you can change into that capital R into the WWR small g or a small g. You should not supposed to take that. Whatever the characters you will take in the first the character, you have to be consider the same traits as the different another character also with the small characters or a small letters. Have you understood now? The second pair of characters is called as the wrinkled greens. Have a look here how to solve this one. Round yellow, when you cross with round yellow with the wrinkled green, you know that round yellow triads are capital R, small r, capital Y and a capital Y, small r, small y and small y. When you cross with these two characters, you will get the capital R, small y, the capital Y and a small y. Whatever the first generation, you will get all the round yellow only. There will be no changes. In a first generation, those characters will dominate the recessive characters or suppressing of another characteristics. What are the another characteristics? Wrinkled green is a completely domination of the round yellows. The first generation you will get all the plants were the round yellow. If you self cross the F1 generation, so have a look. But here we are selecting the pair of contrasting characters. Yes or no? So here we can't we can't solve the same problem as we have solved in the previous class, previous problems. Yes or no? But here have a look, with the help of Punnett box or the checkerboard. You have to write the triads first of all. You have to cross with the capital R and a capital Y, the capital R and a small y. The capital R, this is the capital R, capital Y, the capital R, small y, small r, capital Y, small r and small y. So these are the triads, different triads. So we have to applicable in the Punnett box. Have a look here. The first set capital R and capital Y. Second one capital R, small y, capital R and small y. The third set small r, capital Y. Last set small r and small y. The same way you have to write here also. Capital R, capital Y, capital R, small y, small r, capital Y and small r and small y. So we have to multiply with these characteristics. So we will get the F2 generations, that is the dihybrid cross. Have you understood? While you are calculating this characteristics, have you understood? Be careful. So you might get a confusion with how to write, whether you should write the small r first or the capital R first, or whether you have to write the capital Y first or small y first. So whenever you write the triads, that has to be the capital R or capital Y only. Have you understood now? So this is how we have to write. Again, you have to write the characteristics. You have to assumption, the characteristics of an F2 generation. So there will be 4 different types of the round yellow, wrinkled green, wrinkled yellow, round green. They are the 4 different types. Have you understood now? And count the numbers of round yellow here. The round yellow 1, 2, 3, 4, 5, 6, 7, 8 and 9. The round yellow what? The 9, the round yellow, the 9 characteristics of the round yellow, and next one round green. The round green, have a look at this one, capital R and a small y. Round green 1, 2 and a 3. 3 round green. The next one wrinkled yellow. Wrinkled yellow here 1, 2 and a 3. Wrinkled yellow, there will be no 3. And last only one wrinkled green. The only one characteristics of the wrinkled green. That is 9:3:3:1. It's a dihybrid cross. Have you understood? 9:3:3:1. This is a dihybrid cross. It is the phenotypic ratio of a dihybrid cross. Phenotypic ratio of dihybrid cross. Dihybrid cross. See, that's why the dihybrid cross is very complex, and you have to know, you have to calculate very carefully. And so this is how you will, when it comes to the monohybrid cross, the phenotypic ratio of monohybrid cross is the 3:1. The genotypic ratio of monohybrid cross is 1:2:1. So these are the standard terms of monohybrid cross and the dihybrid cross. And here the most important things in a dihybrid cross, you have to remember the phenotypic ratio. Have you understood now? And I hope you all of you understood how to calculate this one. These are the triads and how to multiply this one. After multiplying, how to analyze the characteristics of each and every triad. You have to be very careful, and once you finish the multiplication of all these characteristics of a triad, you have to write the possibilities of the F2 generation. Have you understood? So this is how you have to do the dihybrid cross. In examination, they will not ask you the same kinds of the characteristics. They can ask you any kinds of the pair of the different characteristics. You have to follow the same rules. The same rules. That could be the easy way to solve the dihybrid cross. See, what are the law of the Mendel's law of inheritances? Have a look here. He is summarized under 3 laws. See, we have before you have learned about the based on 7 contrasting characters. He has conducted so many different types of experiments. Yes or no? The same way, is mainly summarized under the 3 laws. Have you understood? In the 3 laws, law of dominance. What is law of dominance? The law of dominance is the pair of contrasting characters, the present together. The pair of contrasting characters. Which are the contrasting characters? So nothing but if that is a capital T or capital T when it comes to the tall plants, and a small t and a small t, the dwarf plants. The only one is able to express only the capital T, only the tall plants only express their characters. The another one will be suppressed, the dwarf plant will be suppressed, which means the F1 hybrid, the F1 generation, which mainly is a tall plant, that's called as a law of dominance. That is the dominant gene and the recessive gene. The dominant which suppress the characteristics of the recessive genes. Have you understood? So that is the first law of the Mendel's inheritance. The second one, the law of segregation. See, law of segregation which deals about the 2 members of the pair of characteristics. 2 members of pair of characteristics, the separate during the formation of a gamete. See, this is how we will write characteristics. Yes or no? The characteristics, this is how we will write. And when during the F1 generation, we have to write it separately. Yes or no? This is what they are. The gamete, the separate, the formation of the gamete. They do not blend out segregation. The different gametes, they combine together when during the F1 generation only. Only during the F1 generation they will combine together the randomly, the fusion at the time of the zygote formation. The zygote formation which is nothing but that is F1 generation, that the outcome of an F1 generation. Only the fusion of the gametes when for the formation of the zygote, that is the capital T and a small t. This is called as an F1 generation. So in that F1 generation, the completely which bears the tall plants only. Have you understood? And third law of inheritance, the law of independent assortment. See, law of independent assortment, which the distribution of the alleles of one characters into the gametes is independent. The distribution of the alleles of the other character. See, which is nothing but the distribution of the alleles. See, you understood that the dihybrid cross, the distribution of the alleles into the gametes of independent distribution of the alleles of the other characters. When you cross the F1 generation with the self cross, you will get second generation, yes or no? So that is how the law of independent assortment which may be applicable. And the next, what are the importance of the Mendel's law? See, he has conducted so many experiments, he gave us so many theories, yes or no? And he make it in a simplified manner, how to to get the new varieties and everything. So there are some the applications of Mendel's also. In that applications of Mendel's, he gave us the new ideas to combine the different varieties to get the new varieties. In the same way, the great importance for the both the plants and animals, whatever we see nowadays, the hybrid varieties, So all that because of who gave that an idea? The Mendel's. Only he gave that an idea to produce the hybrid plants and also the hybrid animals also. Have understood? That's called as an hybridization. Understood? And the new types of plants, that's called as a hybridization. And the last topic in the genetics, that's a mutation. See, mutation, it is nothing but the sudden changes in the genes that leads in a different ways. Understood? The sudden changes in the genes are a chromosomes or a structure of a chromosome that leads to the different effects. The effect in the sense that leads to the person, the defective. Understood? That's called as the mutation, the sudden changes. In that mutation, there mainly the diseases are there. The sickle cell anemia. The sickle cell anemia, it's a blood disease. Which one? The sickle cell anemia. The causing by the gene mutation, the sudden changes in the genes, which causes by the gene mutation. The mutation causes change in the DNA. The change in the DNA, it results in the production of the RBCs. The RBC shape will changes into the sickle shape. So that's why we call that sickle cell anemia. The next one, the radioactive radiations. See, as you know that during the World War II, in the Japan, there in the US, they have explored the atomic. Yes or no? Atomic explosion when during World War II in 1945, Japan, mainly the two places called as Hiroshima and Nagasaki. Still those people, they are suffering from the genetic diseases. Still they have some defects in their bodies. Not only that, the plants and also the animals, both are they persisting. Understood? So this is the main, the changes in the mutation, so that leads in a different way, that could be the problems. Have understood? So this is about the second our chapter from the genetics, the basic fundamentals. Here we have successfully completed our third chapter in the biology, genetics, it's in the basic fundamentals. Please log on to the global online classes. Thank you.